So it's not like the lactic acid bacteria that were considered probiotic a while ago, but it's the same concept. And I'm a microbial ecologist, and I do have some difficulties to imagine that just one bacterium will completely do the job in this context where it's not just a small defect, it's a systemic defect. So it's the whole systemic symbiosis, as we mentioned, interaction between the human and its microbes that is distorted. And that's what you want to, let's say, repair. And it involves gut microbiota indeed, but also gut permeability, the immune system, oxidative stress, and many other factors that are factors of interaction between the microbiota and organs. And I tend to think that it really deserves a broad, holistic systemic approach to correct the alteration.
So is it fair to say that as a microbial ecologist, you believe that Ackermansia might be an important part of the story, but it would require to be delivered with other elements of the ecosystem to really get...
Yeah, very much. In fact, we mentioned the importance of richness of the microbiota, and we've done some work because Ackermansia is also important in obesity and diabetes, showing that if you have low richness microbiota, you're disadvantaged. But if you have low richness, low Ackermansia, it's even worse, basically. So you would want to think of Ackermansia as an important actor, but it needs to have the right grounds in terms of overall ecosystem, and overall diversity will be very important anyways. And what the colleagues have done actually ever since the initial identification of Acromantia as a player is to try and use testing to see whether they could design a model. So knowing it was more complex than just one species, they looked for a diagnostic or prognostic model. The prognostic model of response or non-response involves 60 different species. Wow. So it's really the whole system that can be modulated, engineered, and that will need to be restructured really completely to potentially hope for a gain in function that would allow response to treatment. In that sense, using fecal microbiota transfer, so the complete ecosystem for reconstruction, for me, makes a lot of sense and probably will be the best option until we have discovered enough, understood enough of the functioning of the holobiont, the human-microbe association or relationship, to be able to fine-tune maybe cocktails. But I really believe that for the moment, for medical purposes, there is a strong sense into using microbiota transfer full ecosystem as a source of reconstruction.
So I think this would be I think that's a perfect segue into you explaining to the listeners a little bit more about the work that you've been involved with in full ecosystem reconstruction through FMT or through the pharma products. That would be fascinating.
Yeah. So actually, I was lucky in 2013 to start interacting with the future CEO of Mad Pharma. So this is a French company that is conditioning human stool to make it a drug, essentially in the context of treatment of cancer for patients who are treated for cancer, for blood cancer, right? And the story began with our will to develop a conditioning system that would be very respectful for the dominant microbes of the human gut, which are strictly anaerobic. So they do not tolerate exposure to air and oxidative stress in no way. So basically, there was a need to condition the material, being very cautious with that respect. And obviously, we wanted to be able to freeze material, to be able to keep it in the freezer for some time, waiting for the patient to need it. And ultimately to be able to lyophilize, which means to turn it into a powder that you can use to prepare capsules, because that would mean an oral form and very simple ways to administer. And so all this path has been done now, but it took 10 years, basically.
It
So
takes a
this
long
is
time. It's
Oh,
hard.
yeah. This
Super
is
hard.
normal drug development, basically, I would guess. And so with Mad Pharma, the first human trial we did, which was started less than 2 years after the inception of the company, was to demonstrate, to bring the proof of concept, basically, that we could reconstruct completely the microbiota of a person that was due to receive a chemotherapy, an induction chemotherapy in acute myeloid leukemia. So it's a blood cancer. And we knew very well that the chemotherapy, which is often followed by antibiotics, and the whole period is 1 month, is completely destructive for the gut microbiota. So the idea was, when those patients are diagnosed, they have not received so many drugs before, so we take their microbiota and we give it back to the same patient. So the donor and the recipient are that one same person, autologous microbiota. And the idea was really to give the proof of concept that we were able to reconstruct.